Yeah, good afternoon, everyone. Again, welcome to another informative session today. Uh, as we all know, and as we all facing this pandemic, this COVID virus, and the entire nation is right now struggling, and we are trying to manage as maximum possible cases as we can. Still, we have seen that Ayurveda medicine, especially the Ras Aushadhis, are having excellent efficacy in terms of managing the COVID patients. And there is one notion, in fact, there is one saying which I always see in Ayurveda, that as far as Ras Aushadhis are concerned, there are very less Ras Aushadhis from South India as compared to North India. There are very less companies in South India who are having good Ras Aushadhis. So in West, in North, we have, but not much in South. They focus on the caste Aushadhis or herbal medicines. But that is not the entire picture. And we have today one of the oldest Ayurveda pharmaceutical company from Chennai, Tamil Nadu. We have the MD, Dr. Narsimmam Jammi with us,
Namaste.
and they have an excellent product that is Pyril DS. It is a Rush Oshudhi, Hutasini Ras, and but not much people, not much Ayurveda doctors and communities aware about this particular product and the ingredient. We personally using in our clinic now from last couple of years, and we are getting very good results, including the COVID patients. We have just ordered few more bottles, and we got very good results in managing. And the main concern is fever. Although the principle is not to suppress fever, the principle is to reduce the viral load so that automatically the temperature will come down. Because if we are forcefully suppressing the fever, the virus will get a favorable environment in our body to grow more, and that's why it is damaging the lungs and it is causing the pneumonitis in many cases, and it has been now researched and published as well. Still, people are skeptical, and as soon as they see anything above 100 in their thermometers, they start taking antipyretics and suppressing the fever. So, what actually the Ayurveda medicines can help? How much effective in terms of controlling the fever, controlling the temperature? कैसे हम इनकी help से we can manage the main important aspect, which is the temperature which is rising and simultaneously reducing the viral load. So without wasting much time, let's understand all these concepts, and let's understand about this very less, I will say, known ingredient and known medicine way from South India. Uh, Thank you again, Dr. Narsimham Jammi. He is a learned professional, a very well experienced person, and the best part I personally liked about him is he has a synergical mindset. The synergies he has uh, like infused, the mindset he and his company is infusing and imparting is one side the traditional knowledge, the know-how we have, and the other side the modern science, the latest scientific parameters on which we can evaluate, and we can have the all the Ayurveda medicines being positioned. So that is the best part, which is I think the need of our now. So over to you, Dr. Jammi, for the entire session, and hope we all will understand more about this particular medicine. And I request all of you who are watching us live right now, as you all know that we are having a limited availability on Facebook, but we are very much available on YouTube. Right now, this session is going live on YouTube. We'll be sharing the video link with all of you so that you can share with your friends, seniors, and juniors after this session as well. Meanwhile, if you feel any query, any question, you can post in the live chat, and we will be able to answer after the talk and presentation of Dr. Jammi. And you need to connect with the company. I request all of you to share your details, your name, number, and email ID. They will love to connect with you and provide you the samples, and accordingly they can take it forward. Thank you. Over to you, Dr. Jammi.
Yeah, hi. Thank you, Dr. Piyush. It's an honor being here again. It's been a while. Um, so actually, I want to talk to you about talk to all of you about one particular aspect of Ayurveda, and you are all experts in it. My background is slightly different. I come from a Western science perspective. My PhD is in molecular biology and biological chemistry. My postdoctoral work in Stanford was in molecular pharmacology. So I understand toxicology, I understand pharmacology very well. I then switched over to Ayurveda only about 12 years ago, 13 years ago. I learned from my father, who learns from his father, and so on and so forth. So my advantage, or I should say, my the best part about my training is that it's very practical. I understand a lot of things the way my father taught me, and I think it's a very uh, practical way to go about practicing Ayurveda. And so to make things very clear to everybody here, is it's I'm a scientist at heart. So my passion is for science. My passion is to understand how things work. And long ago, when I was doing my PhD, people started questioning Ayurveda, and I come from a family of 125-year-old legacy of Ayurvedic Vaidyas. And my father himself is an allopath who converted to Ayurveda, and I've converted to Ayurveda. And so my question was, if Ayurveda was so bad, and if Rasaushadis were so bad, how was it that we've stood the test of time for millennia? There has to be some rationale, and there's got to be some way in which we can understand this. And everybody's like, it's so toxic, it's a heavy metal, how can you use mercury, causes all kinds of the birth defects, the Minamata disaster in Japan and all that. But what people really don't understand, and what people seem to forget about it, is that there's a Latin saying, sola dosis facit venenum, meaning it's the dose that makes the poison. Okay, this is the central concept of toxicology. And what this actually means is that toxicity is entirely dose dependent. For example, you know what? There's a concept called LD50, lethal dose 50, right? So the lethal dose 50 for the most important thing in our lives, water, is 6 liters. Okay, so if you take 6 liters of water in one shot, you will die, right? Because you get hyponatremia. Now, you can even take small, very extremely minute doses, in just minute doses of snake venom, you won't die. So what's the difference? On the one hand, you have a known poisonous substance. On the other hand, you have the water of life, the elixir of life. Yet, if you take it beyond a certain point, you will die. And if you take very minuscule contents of snake venom, you're not dead. This is an established fact. This has been done in many studies. Now, here I'm going to talk about a very interesting compound, Ayurveda's most wonderful antiviral. I'm very specifically talking about antiviral. I don't want to mention the names of the disease that we're tackling here. Everybody knows we're talking about the disease that's really becoming a part of everybody's life right now. And the reason why I'm not using the name is because we're getting banned in Facebook. We're getting Facebook. The other day I put up a post on Facebook about Ayurveda and COVID, and I was given a warning. And obviously, Indian Vaidyas has been banned just because we advocated Ayurveda to cure this present pandemic. So I don't want to mention the name of the disease, but you all know that's exactly what I'm talking about. And that is what this is when I say antiviral, you can all assume that what I'm talking about is the present situation we are dealing with. Okay, now this particular medicine that I'm talking about, Hutasini Rasa, is a very unique one. It's a very rare rasa. Not very many people make it because it's a very elaborate process. And actually, if you stick around at the end of the presentation, I'm actually going to show you a 5-minute video on how we make it. We've actually taken it out away from our manufacturing area so we could get the right light. I'm going to show you a video of the entire process of making Hutasini Rasa. And Hutasini Rasa is a mercury-based preparation. It's a rasaushadhi. It's based on making, first you make kajali, and then you make add a whole bunch of things. And I'm going to show you everything. I'm going to walk you through it step by step. Okay. And we use pure mercury, and pure mercury is actually very, very, very non-toxic. Surprisingly, it's the inorganic mercury. It's the mercury chloride. All of these compounds that actually are very toxic. But if you take pure mercury, like quicksilver, parada, as I say, the one that actually is shiny, liquidy stuff, that's actually not very toxic. We're obviously not using it as it is. We're using pure mercury as a start, and then we're going to go ahead and go through a whole bunch of processing. It takes a week to make Hutasini Rasa. It's also known as Hutasana Rasa. It depends on which textbook you see, but we are going to use Hutasini Rasa because that's what I've grown up with, and that's what I'm used to doing. So without further ado, this is the background. I'm going to start my presentation, and I'm going to share the screen. And just 1 second while I do that. Okay, I hope you're all able to see the screen right now. So we're going to talk about Ayurveda that's relevant for the post-pandemic era, and I'm going to go through a series of scientific experiments that we've done, because for me it was a challenge to actually show that Hutasini Rasa is not toxic. It's actually efficacious and actually very safe. It's not just non-toxic; it's also very safe. Now, Pyril DS, as we call it, is in our experience. You know, we have a clinic here in Chennai, and we have cured hundreds and hundreds of patients with various viral diseases. And lately, with the present pandemic, we have also worked very effectively. It has worked very effectively along with a whole series of other medicines that we give. I'm going to show you the treatment regimen also at the very end. Now, this is a knowledge sharing talk. This is not a talk. This is not a marketing talk. This is a knowledge sharing talk. We want to make sure that every Vaidya who's seeing this, every doctor who's seeing this, understands and is able to treat COVID patients, or the present pandemic patients, because we want people to get better. We want India to get better. We want to get going with our lives. And Pyril DS is the indications as per the textbooks is it's used in dyspepsia, indigestion, coughs, colds, viral fevers, as well as fevers of unknown origins, and then bronchitis as well. Now, what is this? We actually then I'm going to go through all of these scientific experiments in very a basic manner, so you actually understand the experiment we did, and how we are showing that Hutasini Rasa or Pyril is extremely safe. Now, there is an assay called Mtt assay, right? And there are two graphs that you see. One is in black and white, the other is in green. These are two different cell lines. One is a cell line called HepG2 cell line. The other one is called a HER7 cell line. What these cell lines are not that important. One is a liver cancer cell line. The other one is also hepatitis carcinoma cell line. Now, the reason why we use this is because these are very standard cells you use in cell biology to actually test the toxicity of a substance or the safety of a substance. Now, we use the cell line called Mtt assay, and we look for the cell viability, meaning after adding the drug, whether the cells are still alive or they start dying. Okay, so this is the very basic step any cell biologist will take to go ahead and do the safety efficacy study. So this is the safety part of it. So if you look at it, the dark gray circle on the left, bar on the left is the drug, which is Hutasini Rasa Pyril, and the light blue is a control where you don't actually add any drug to it. If you look at it, both of them, the cell viability is 100%. It has not done any damage to the cells. Now, if you look at the Mtt assay on another cell line, which is actually a very finicky, very sensitive cell line, which is why we took the one on the left is a very rugged cell line, so it can withstand a lot of rigor. But the HER7 cell lines are actually a very finicky cell line, and we wanted to see how these two will work in a very sensitive cell line. Okay, these are liver cancer cell lines, and here also we show that whether you add the drug or whether you don't add the drug, the viability of the cell lines are very, very similar. Okay, if the dark is the one with the drug, the light is one without the drug, and they're two different concentrations of the drug. So basically, the take home message of this graph is that Pyril DS is a very safe medicine formulation to take, which does not lead to any cell death in experimental conditions. And we also so it doesn't lead to cell death, but does it actually allow the cells to grow? So that is also important, right? It's not just important if it's not toxic. We also want to make sure that it's safe. So there are two different aspects to this whole concept over here. So what we've done is we've actually taken Pyril at various different concentrations. If you look from left to right, left is the highest concentration, which is 1000 micrograms per ml, and the one at the most at the rightmost, the orange bar, is the one without any Pyril added. And what we do is we treat these cells with Pyril. We let them grow for 48 hours. And then we start looking for the number of cells that actually died. Now, please understand and appreciate that 1000 micrograms per ml is such a high concentration that even if you add 1000 micrograms of ml of let's say sodium chloride, which is salt, or of almond extract, anything, think the cells will die. So the 1000 micrograms per ml is actually done to show at what up to what level you can resist the toxicity of an added substance. Now, if you actually see it, right, so you start looking at it, you see that, even at very high concentrations, even at 1000 micrograms per ml, there is no difference. You look at the error bars, you see those little T-shaped bars over there, they're called error bars. And if you look at it, it is well within the range of error. So even at 1000 micrograms per ml, Pyril is allowing for the cells to grow. Okay, so the next thing we wanted to do is we actually wanted to look at the amount of mercury that's present in the in Pyril in this particular formulation. So we did something called atomic absorption spectroscopy, which is a very sensitive method wherein you can actually detect that particular element. So there are standards that you can use. You use this, you actually look at the particular, you run a control, you find out what is the signature of that particular control. For example, in this case, it's mercury. So we found that once formulated, mercury was below detection limit. Now, this has not come as a surprise to me at all, because what we expected that you're not going to be able to see inorganic mercury in this. That's exactly what we expected, because mercury has gone through such a long, laborious processing, a Shodhana process, which I'm going to show you as we get closer to the talk. So where we've seen that, it is not a surprising fact at all that this is actually not able to detect mercury in it. So which means that mercury has undergone some sort of a change wherein it's not being detected by atomic absorption spectroscopy. Now, the basic ingredients of hutas. Now let's get to the Ayurvedic aspect of things. Hutasini or Hutasana rasa, like I said, has got purified mercury. It's got purified sulfur. I'm also going to show you how we purify sulfur. It's got purified nabi, and then it's got kushta, triphala, trikattu, bringaraj, and ginger juices, right? And then this is the preparation. We use Yoga Ratnakara as our guiding light to how to go about making Hutasini rasa. We follow this. Here we use here it is if you look at one particular section here, it says you have to use Griha Duma. I don't know how many of you actually know what Griha Duma is. Griha Duma actually means cobwebs. And since we can't get cobwebs right now, we actually use burnt coconut shells. The reason for using burnt coconut shells is because it's a great adsorbent, gets rid of a lot of toxins from the body. So that's what we use. And Grihaduma, in my great grandfather's days, I actually heard stories. I was not there when he was born, when he died. But what I heard stories was that they would go from house to house and collect cobwebs. But since cobwebs are no longer a feasibility at this point in the modern age, he laid down rules, and my great grandfather said you can use coconut shells, burnt coconut shells, and that's what we use right now. Okay, so now I'm going to show you the Hutasini rasa preparation. I will walk you through each step. There are some steps that is going to take a long time, so I'm just going to show you a fast-forwarded version of some steps of the process. Now, I hope you can see this is purified mercury. You need a license to procure mercury. That right here, what you're seeing right here, that is actually cow dung. Okay. Now, please remember this. Ayurveda cannot exist without cow dung and cow urine. Okay, and it's impossible for you to be able to formulate something if you actually don't use this. Now, and again, it's very important. It's not just any Jersey or Holstein cow or whatever it is. It is actually cow that we need to get the cow dung from an Indian desi cow. Those are the ones that have the medicinal properties. Okay. Again, the Gomayam, as we call it here in South India, has to be collected during Brahma Muhurtam. The very first cow dung that the cow passes in the morning at 4:30, between 4:30 and 6. That's what we have to collect. Right. So there are rules for it, and this has been handed down to us from generation to generation, and we don't change anything. While we may be very modern in our outlook towards the science and the the way we go about understanding it and analyzing it, when it comes to preparations, we do not cut corners. We do not make any changes from what my great grandfather has laid down as rules. Right. So you basically dissolve this and you add the mercury to the cow dung, and you let it sit for 48 hours. Okay, 48 hours it has to marinate in this cow dung. Okay, now you can actually see it, and then you wash off the cow dung, and you go through several washes. Right, this is you wash it and wash it and wash it and make sure that all the cow dung is gone. So I'm not going to show you all the washings over here. I'm going to show you make sure that you can see up to the point where we finish washing the process. And then what you do is you filter it. You filter it using a muslin cloth, right, to make sure that after this is after several washes. I think what we've done is we've done about 25 washes with distilled water to make sure that all the cow dung is washed away. But remember this, before it was done, it was marinating in cow dung for 48 hours, 2 days, right? And then what you do is you actually squeeze the water out of it. When you squeeze the water, you actually see that the mercury also comes out. There you go. You can see that mercury coming out. You squeeze it very, 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 very nicely, right? So this is the Shodhana of mercury. Okay, this is how mercury is purified. Pure mercury, as in inorganic pure mercury, is purified. Okay. Now, again, there has been no chemical change at this point in time. We don't know whether there's been any chemical change at this point in time. We are undergoing. There's a lot of studies being undergone where post. Now, this is here is Nabi. I'm showing you how we purify Nabi. We actually put it in an earthen pot. And we use again the milk that you see on the right hand side is non-boiled, okay, A2 milk from a desi cow. Okay, Nabi is soaked in milk, and this is done with a daily change of milk for 7 days. So please see this, it's completely soaked. All the entire Nabi is soaked. Again, you can use either Nabi or you can use Gomutra. Okay, one of these two can be used. Again, if you're going to use milk, it has to be desi milk. You don't use Jersey Holstein. Again, if you're using cow urine, it has to be desi cow urine. Okay, so this is something that we do. Now, here, what is the next step is the Gandhaka. This is sulfur. This is again raw sulfur. And please take a look at how it's being purified. Now, you take embers, glowing, burning embers, you place it on the sulfur as it is. And previously, if you showed, there's milk in it. There's again desi milk in it. And what happens is that as it gets heated, the sulfur melts. And when the sulfur melts, you see the mesh over here. It passes through the mesh as very small balls. Pretty much all Ayurvedic companies prepare sulfur only this way. This is the standard classical way of preparing sulfur. Okay, so you start seeing that it starts falling down into the milk, and then you let that sit for overnight, and then you nicely do the litigation of it. You nicely do it, make it into fine powder. Okay, so this is sulfur. This is as far as sulfur. The next important step is the addition of the purified mercury to this Gandhaka. So the Parada and Gandhaka together form Kajali. Now look at one is nice, beautiful yellow in color. The other is like silver color. When you actually put it together and you actually mix it and you grind it and you grind it and you grind it, you end up with Kajali. Kajali is a dark black substance that is actually very, very useful for all of us in so many different. There you go. That's the black powder that you see. That's Kajali. That's the combination of mercury and sulfur. Purified mercury, purified sulfur. Remember, both of these things take a long time to purify. So the entire process of making Hutasini Rasa takes a very long time. Now what you're seeing here is lemon juice, right? It's lemon juice for a very small batch. What you're seeing now is about 10 kilos of Hutasini Rasa. We squeeze 200 lemons and get the juice from that, right? So we don't use citric acid. We actually need to use lemon juice in its original form. So we add it, and then we make sure that you can see the lemon seeds and everything. So you make sure that you do the grinding, you do the litigation for a very long time. We do this over a period of 24 hours. People take turns because it needs to be done. People's hands start hurting. So this needs to be done in a very, very methodical manner. And eventually, then you take that out, you dry it, and then you tablet it, and that is what we use as Pyril DS. Now, as you can see, it's a very laborious process. It takes a long time to go about doing it, and we use this as part of our treatment regimen, right? The treatment regimen in Jamni's clinic for the present pandemic situation is as follows: Pyril DS, 2 to 3 tablets. 3 times a day. If the patient is below 60 kilos, we give 2 tablets. If they're above 60 kilos, we use 3 tablets. We recommend that you use the same. Please use it 3 times a day. Nothing wrong. As you can see, the dose is 100 mg. Each tablet is 100 mg, right? And there are so many other things that go into it. There's Tricatu, there's Triphala. So the total amount of quote unquote heavy metal that you're worried about is very minuscule, but that is so important in being a potent antiviral for today, for today's situation. Then Sudarshana Churna capsules, we've capsulated Sudarshana Churna so that because it's a very bitter medicine, but there are 48 different ingredients in Sudarshana Churna. Again, one of the greatest immune boosters known to Ayurveda. 2 capsules 4 times a day. Immune Zen granules, 1 spoon, twice daily after food. Then Kabasara Kudineer, which is a Siddha preparation, which we use as a kada. We ask people to make it at home, freshly made, 30 ml twice a day. AA caps, 2 capsules twice daily. Now, if the person has a cough and a sore throat, the same 2 capsules are to be emptied into a katori, pick a small katori, take 10 ml of swascoline syrup, mix well, nicely mix it well, and then consume it 3 times a day. And this is for people with once the lung gets involved in the whole process, the cough and sore throat and all those kinds of things. When you have a spasmodic cough and all that, you need to use this particular regimen. You do this, and we've started seeing a lot of improvement in a matter of 4 to 5 days. People start getting better, but we tell people they have to quarantine themselves for the entire 14 to 15 days, whatever it is, until they test negative in the RT-PCR test. So what I'm going to talk to you about is a 51-year-old male with a mild, he started off with a mild disease progressing to a more serious case. I want you all to pay attention to how methodical and how diligent he was. He had, please listen to his this experience, and I want you all to listen to it. So please just give me 2-3 minutes here.
Sir, voice is not coming.
Oh, is it? Okay, let me see if I can increase the volume. How about now?
No, actually earlier it was coming, but now there is some issue in the voice.
Okay. So if it's not clear, I'll try to explain it. Shall I
What I
just say it?
suggest, what I suggest is after this event in the YouTube video comment section, you can post any link of this testimonial.
I will do that. I will do that. So basically, this was a patient with very severe symptoms, and they actually took this person. He refused to go to the hospital. He refused to take any allopathic medicines. He only decided to cure himself with Ayurveda. And you know what? This is what I showed you before is our treatment regimen, right? It is not. It is not. Every doctor has his own treatment regimen. But what I am saying is, please use our expertise. Please talk to us. We are happy to share our experiences with you, our treatment modalities with you. Please feel free to use your own experience and your own favorite rasayanas and your rasaushadis, whatever it is works for you. But since we have a lot of experience with our regimen, and this is something that we've been actually giving it to a lot of patients and a lot of things, we actually have come up with an immunity boosting kit where we give all of the medicines I spoke about, and these are things. Yeah, sorry. So that's the um, end of the slideshow. And so what I actually want to tell you was that we will share everything with you. We want to make sure that you all get benefit of this particular treatment regimen. And feel free to reach Mr. Madhusudan. His numbers are with you. In case you don't have it, let me just give you his number. He's our head of marketing, and he will be able to guide you to the various ways in which you can procure these things. Let me just give you his number right now. I don't have it on top
I
of my head.
suggest Dr. Jamil,
Shall I type it?
first of all, thank you so much for this informative session. And I think all of us should focus on Ras Aushadhis as far as managing and combating this deadly virus. And I request after this session in the comment box, because this is the live chat, so people is not able to capture that. What I will do is after the let me share right now here as well. But after this session also, in the comment section box, we will post contact details, and all of you can connect. There are a couple of questions. One is the dosage. So as far as the Pyril DS is concerned, you maybe just let everybody know. And second is many Rasoshadis, almost all Rasoshadis are coming up with Anupan. Like we have to give it like for example mixing it with honey or adrak swaras or gogret or that kind of thing. So maybe you can clarify on these two aspects, like dosage also. There are three categories which we need to understand right now. One is the mild stage in which the temperature is not that much, another is the moderate, and a third one is the critical in terms of the temperature is concerned. Like mild means 99 or 100 maximum. Then there is a second stage where people are having 101, 102, and many are having more than 102 as well, 103 and 104. So what kind of dosages we can administer in these three category of stages of fever? And second is the anupan. So please clarify on these two aspects.
Okay, so the Anupana for the easiest one, the Anupana for Pyril DS is milk. So please drink as pure milk as you possibly can. Now, as far as dosage is concerned, for mild, so for all mild, medium, mild, moderate, and severe, it is two tablets 3 times a day. However, it's the number of days that you take it that's going to be different for the mild versus moderate versus severe. Mild, we tell people that until the fever is coming down, you make sure that you don't have fever. Take it for one more day after you have no fever, and then stop. Moderate, you take it whether you think you have fever or not. We ask people to take it for 1 week. For people with a very high fever, we tell them to take it for the entire duration of for as long 15 days, and then one more day after they've noticed that they have gone for 24 hours without a fever. So it's a very standard, very straightforward way in which we tell people to take Pyril DS, 2 tablets 3 times a day. Based on, it's a weight based thing. 2 tablets 3 times a day if you're less than 60, if you're less than 60 kilos. If you're more than 60 kilos, 3 tablets 3 times a day. The duration is what is going to change. So if you're a very mild disease, 3 days you can stop. Make sure that you don't have fever for 24 hours, and then stop taking it completely. 7 days if you're moderate, make sure you don't have fever for 24 hours, then stop taking it. For severe cases, continue taking it until you're completely all right, your RT-PCR is negative, then one more day after you're completely negative, and then you can stop. That's how we've been treating our patients.
Right, and one more quick question, and then maybe we can conclude the session, as all the practitioners are right now involved and busy, and also we want to have a crisp, short video so that everybody can watch later on as well. But one of the most important question of these days, again, I would not like to name the disease, but treatment protocol. As far as your team, we know that you have great footprints in Chennai and that part of the country and in the entire South India. So many people are following, and most of them may be using your clinic treatments or your company treatment protocols. So there are some differences also. I know that as far as your pharma company and your clinic treatments are concerned, I'm not much aware, but I know there are some differences as well. The question is straightforward. As far as the COVID protocols are there, as per the Ministry of Ayush, they have shared certain preventive measures and all. Then there are allopathic medicines protocols going on, and so there are some confusions as well, whether to take it with allopathy, to take it standalone, only Ayurveda, or it is like integrated along with allopathy we can take. What about the people who are on oxygen support, critical hospitalization? Although we have done several sessions, still we want to hear from you. What exactly you people are doing? What exactly your clinics, your company, your doctors who are associated with you? So maybe in next 5 to 10 minutes, you can just explain briefly about the entire aspects which are there. We have also heard there is one new deadly strain of Andhra Pradesh, AP strain, which is
Yes.
causing much issues. We are not aware about any other strain. There is a Tamil Nadu or Chennai, but maybe in a nutshell or in a crisp next 5 to 10 minutes, you can share these entire aspects with all of us so that all of us can get benefited and accordingly we can take it forward.
Yeah, the AP strain is the new one where people have started getting low oxygen content in a matter of 3 to 4 days. Actually, it was first found in Vizag, and we urge people in Vizag to be very careful. Now we are very clear when it comes to allopathic treatment. We tell people, you know what? So most important thing that we've noticed is this fear and this anxiety and this worry. Whether you're in the best of care, the best of medicines, if you're going to be worried and if you're going to be so stressed out, none of these medicines are going to work, right? So that, because the stress is such a silent killer, and it makes sure that you're not your immunity is going to become low if you're completely stressed out. So In order to assuage people, in order for people to be very comfortable, we tell people, please take the allopathic treatment. Go ahead. What is there in the allopathic treatment? There's nothing. There's like, you have paracetamol, you have azithromycin, you have vitamin C, you have zinc, you have bicosils. I mean, this is what the doctor is giving them. Please take it. There's nothing that is only going to make sure that your fever is suppressed. It's not going to get rid of the virus, right? Now, the azithromycin is given because this new strain has started causing a lot of sore throats, secondary infections, which is why the azithromycin has been given for this new one. In more severe cases, people are getting steroids but those are all people who have been hospitalized and where they're having very high inflammation. As far as oxygen is, when they're on oxygen support, there's no question of us being able to give them these medicines in the hospitals. So that is ruled out. We tell people that if you're in a hospital, there is no way you can't even sneak it through. They don't even let you go inside the hospital. So how are you going to give the patient the medicine in the hospital? So this is for people who are at home. If you're not in the hospital, I actually think you're very lucky. I actually think that if you're not in the hospital, it's good for you because you're able to take good care of yourself. You're not exposed to a lot of different people. You're in isolation. If the allopath has given you a set of medicines, please take that medicine. It's not a problem. It is not going to bother because the two systems are orthogonal to each other. When I say orthogonal, one doesn't cross-talk with each other. So the Ayurvedic treatment, any Ayurvedic treatment does not cross-talk with the allopathic treatment. We are very clear on that. Please take your Dolos and your azithromycins or whatever jo bhi lena hai, please take it. It doesn't matter to us. But make sure you follow the dosage that we give you, the regimen that we give you, and we assure you that you will be fine. You will get much better in a matter of 7 to 15 days, depending upon the severity of the disease that you have. That's all I have to say.
Right. Thank you so much, Dr. Jammi. I think this is the mindset we all need to understand. Although you are absolutely right that they are not allowing any medicines in hospital, and there is a challenge, but still we were able to manage, and few of the patients and several other doctors are also trying, and few patients who have received the medicines in ICU or with oxygen support, we have seen very good recovery. It acts as a catalyst. So what I suggest and what I like right now, we are suggesting to all the people who are connected to us, see that two aspects we need to understand. First is there is nothing which will prevent this virus to enter to your body. So there is nothing like, okay, your immunity can be good, you can fight better, you can fight without hospitalization or without oxygen also if your immunity is good. And maximum people are fighting like that, 90 to 95%. But the problem is the remaining 5 to 10% are in risk, and there is no tool, there is no test that determines, okay, this person will not go into the critical hospitalization stage. You never know that. People with good immunity, people with young age are also going that. People, doctors, Ayurveda doctors, allopathy doctors, many people are getting critical hospitalized, and so many casualties are there. So there is nothing which will help in preventing this virus in entering your body, and also there is nothing which helps in killing at a very effective or at a speedy way. So this will do the harm. The only thing which you need to understand is, as far as Ayurveda medicines are concerned, they are specifically they have started reducing the viral load immediately as soon as it has been administered to you. Second is the damage control, if that is very fast. So we need to have some critical care and emergency medicines which are there in our counterpart system, that is allopathic, in terms of steroids, in terms of oxygen support, ventilator support. So damage control should be checked and should be stopped immediately. And if along with that Ayurveda medicines are being prescribed and being administered in ICU and oxygen support, I request all the fellow physicians, allopathy doctors also, if possible, please allow, because we have seen those who are getting, their recovery is very fast, because ultimately people are getting recovered with the support. There is nothing which is helping them. It's their own immunity and their own willpower and their own system which is getting reversed. Yes, we can support them, we can delay, and we can control some kind of a damage control. But if damage control has been done, for example, we have seen in HRCT chest report the lungs is being damaged of a score of 15 or sometimes 20, about 25. So the steroids and other allopathic medicines can uh, stop for a while further damage, but Ayurveda, if infused at correct time, will reduce that score to maybe less than 10 or sometimes less than 5 also. We have seen in many cases within 1 week it has been done. So that is the ideal approach we all should understand, and we all should, without see, right now there is no point in proving which path is superior and which path is inferior. I think
Yeah,
we
that's
need
irrelevant.
to
At this point in time, we need to save lives.
save lives. So if we can integrate and if we can amalgate both pathies right now, that can be the best possible way to manage and to prevent more and more deaths. I think with our limited exposure and with our limited knowledge, this is the only suggestion we want to give to all of you who are listening to us right now. And thank you again, Dr. Jammi, for this exciting and
One more
informative.
important thing. One more important
Right,
thing.
right.
Please get vaccinated. All of you.
Absolutely, absolutely. And in
Make
fact,
sure
we
that
have
you get vaccinated.
absolutely. We have seen that the criticality and the lethal criteria is getting significantly reduced in people who are getting vaccinated. Although there are some risks which I want to clarify here, and we want to ensure that all of you are updated after getting vaccinated. Do remain at least for few days in your isolation, because you can be a potential carrier also. We have seen many people immediately after getting vaccinated are having fever and some symptoms, and their reports are also COVID positive. So maybe due to the antibodies, one, maybe some people are having their immunity less, so they are getting as a COVID carriers. But whatever, so you have to be remain in isolation for few days after getting vaccinated. Although almost all the states are in lockdown, so you are only supposed to go to a vaccine center and then you have to come back to your home. So there is no virtual, there is no practical aspects of getting. In fact, you can't roam around in your societies also these days. Many societies are sealed
Correct.
for walking in the premises and that kind of stuff. So you have to be isolated, and then only we all will be able to fight with this deadly virus. So thank you so much again for this exciting session. And I request, many doctors are posting their queries, and they have posted their numbers also. So I request Mr. Madhusudan and the entire Jammi team, please connect with these doctors and provide them the tools which are very much required right now to fight this
I'm not
infection.
able to see any questions, Dr. Piyush. Is it because we are on? It's not being streamed here, or is it all being shown on YouTube?
Yeah, so it is there on YouTube, and many,
Okay.
actually
Okay.
this telecast has been broadcasted on our website page also. So from
Okay.
website page there is a direct inbox which we are getting the questions. So these few questions which I have suggested, we got and we requested to you as well. So we are
Okay.
having these comments from these let me just display on the screen now. So
Okay. So it's all on YouTube. Understood. Understood.
yes,
Yeah.
yes.
Okay. Got it. No problem. We'll look at the YouTube session, and then I will try to answer as many questions as people have. Not a
Dr.
problem.
Sushma is asking how to get the medicine.
Yeah. Yeah. Mr.
Dr.
Madhusudan will take care of the information. Absolutely.
Dr. Snail is also like requesting for the details.
Yeah.
So I think Dr. Sushma is in Chennai, so you people will be able to help her
Oh
as
yeah.
far as
Absolutely. Super.
right.
We'll do it.
So
We'll take
she's
care of
in
it.
Annavaram, Chennai 12. Right.
Ainavaram. Ainavaram. Yeah. Yeah. We'll take care of it.
Dr. Snehal is in Maharashtra. So I request all of you, and see, this video will be now broadcast to all our followers who are connected to us. So later on, maybe this evening or night, they will see. And I request all of you who are watching this video, please post your query in the comment section, and the team from Jammi will be able to help you, and we'll be very much happy to answer your queries. Thank you so
Yes.
much, Dr. Jammi,
Thank
for
you so
this
much.
session, and take care and have a great week ahead.
Namaste. Namaste. Thank you all. Thank you.